story I'll tell you about will, I think, alter the way you interact with a common product this evening. So that's the suspense for you, okay? So you all know this famous picture. You know this famous picture? Yeah? But you haven't seen this picture, actually, because this is the one before, it was taken before the iconic photograph. And do you see what Crick is doing? He's got his hand round the DNA backbone over here, right, with two fingers round it. And actually, inadvertently, he is repairing DNA. But of course, he wasn't doing it in anticipation of an important field of biology, but because he was actually wanted to take an iconic photograph, which is with the DNA strand fixed. Okay? So the story I'm going to tell you is about DNA repair, how you maintain genetic integrity, and why it is fundamental to maintain genetic integrity. And I'll walk you through a kind of detective story of how we got to what we discovered. So why should anyone be interested in DNA repair? And this is some work I did as a postdoc centuries ago. And what you see here is a cell. I'd made a cell line from a mouse that lacked BRCA2. Now you all know what BRCA2 is. 5% of hereditary breast cancer is due to a mutation in this gene. But at that time, nobody knew what it did. But I made a cell line from a mouse that lacked BRCA2. And what you can see in this cell line is that just by putting these cells in culture, what I noticed was that the chromosomes spontaneously broke. You see these? Yeah? So that's a normal mouse chromosome, and you can see that the chromosomes have snapped. And what this observation told me was what I set myself up to do for the rest of my life, really, was to, what it really told me is that by merely undergoing cell division, there are factors that operate within a cell that cause the DNA to break. And so it's fundamental to fix this damaged DNA. So there must be factors that damage the DNA naturally that occur within us, and then there must be very powerful systems to fix this damaged DNA. And this really shows that BRCA2 is an important component of that fixing process. And why is this important? It's important because actually the majority of cells that have this kind of damage don't ever survive, they die. But a few of the cells do go and survive, and what you wind up is what's shown over here. And so this is a painted image of chromosomes from a woman with BRCA2 breast cancer. And what you can see is that each chromosome should be a single color, but you can see that there are many chromosomes with hybrid structures, yeah? Well, translocations. So the story here is there are natural factors that damage DNA. There's powerful mechanisms within the body to fix that damaged DNA. And if you don't fix it, cells die, or you wind up with translocations that drive cancer. So it's a fundamental root of developing cancer. So in approaching this problem, I began through human genetics and a very simple idea and concept. And we turned to rare human genetic illnesses where this whole process of fixing damaged DNA goes wrong in the first place. So I'm going to introduce you to a very rare and unusual disease, but it's got a remarkable phenotype, and it's called Fanconi's anemia. Feel free to interrupt me if anything is unclear. So you're born with a genetic mutation in the ability to fix damaged chromosomes, and the consequence of this is that the children get developmental defects, shown over here. You see that this child's hands are very abnormal. But if these children survive birth, then they lose their blood stem cells, and they die of bone marrow failure. And if they survive that through a bone marrow transplant, they get cancer. And the risk of getting cancer with this genetic disease is about 10,000-fold over wild type. So does anyone know what the risk if you're a chain smoker for lung cancer is? Fold difference. Anyone? 25%. That's a very powerful word. Do you mean 25% of all chain smokers get lung cancer? It's not true. No. Quarter of chain smokers? No. It's a tenfold above normal risk, right? So this is it's about 1,000 to 10,000-fold, okay? Which basically means that every child who has this disease gets cancer. In fact, we know why they get cancer, because they break their chromosomes, and the clue of why they break their chromosomes is they get an unusual defect in DNA. So there is a factor out there that causes this thing, which is a chemical agent that glues the two strands of DNA. And so you have to separate DNA in order to copy it. So if your two strands of DNA are glued together and you don't remove this, then what actually happens is you wind up with shattered chromosomes. Okay? This agent is called an interstrand crosslink. So we know that there's cellular defect in this disease. But there's another unusual feature of this disease, and this is a very busy slide, and all you need to take away is that every family with this disease has a mutation in a different gene. So in a way, you will have heard of genetic screens, yeah, in flies and mice and so forth. In a way, nature has done a genetic screen for us for factors that repair crosslinks, DNA crosslinks. And these are all the genes that have so far been isolated when mutated in a homozygous configuration lead to this disease, Fanconi's anemia. And a couple of interesting, there are in total about now 17 genes, but you will see this group over here, and that's actually BRCA2. So in fact, women who carry one bad copy of BRCA2 don't get Fanconi's anemia because they have the good copy to rescue them from, right? But if you're unfortunate as a child to be born with two bad copies of BRCA2, you get this condition that I've told you about, okay? But really the key question in the field was, why did you need to fix these crosslinks? How did the accumulation of these crosslinks cause problems of development, make your stem cells disappear and give you cancer? And so if you're not chemists, the very simplest way to look at this problem is what I've shown you here is the standard cross-linking agent. Does anyone know what that molecule is at the top there? Yes. Has anyone heard of cisplatinum?
Yeah, of course.
Yeah, that's cisplatinum. So what do you use cisplatinum for?
For treating cancer.
Yeah. So you use it to treat cancers because cisplatinum glues the two strands of DNA very efficiently together, yeah? And that's why it kills cells, because it saturates the system to remove these crosslinks, okay? And in fact, if you lack this Fanconi repair pathway, you're extremely sensitive to cisplatinum. You can't give it to these individuals should they get cancer because all their other cells just fall apart, okay? But obviously, evolution can't have selected for a repair pathway conserved all the way down to very simple organisms based on the fact that you're going to be exposed to cisplatinum, yeah, which is a man-made molecule, okay? There must be a natural factor that's causing this. And it's the identity of this natural factor which we discovered some years ago. And I'll tell you straight away what these two natural factors are, and it's these two molecules. So who's a chemist over here? Do you know what these are?
It's written.
Sorry?
It's written there.
It is, yes. Do you know what an aldehyde is? An aldehyde is an organic molecule with a terminal carbonyl group, yeah? This group over here, right? And where do you think the aldehyde comes from in you?
Well, from metabolism.
Yes, and
Byproduct.
and what else? I think Czechs are quite keen on it.
Alcohol.
Yes, and that's the connection. So this goes to show that we first screened for these molecules, and we bought them from a chemistry shop, and then we dumped them on our cells that lack this repair pathway, and you can see that the cells shown in red over here are rapidly killed by this chemical, okay? So, well, that's all well and good. How can we show in the physiological situation that the amount of these natural molecules you produce within you are enough to cause this kind of damage, necessitating repair as well, repair? So for this, I need to tell you where these molecules come from. Your colleague has already told you that they come from cellular metabolism, but they will also come from the glass of gin gin and tonic you have tonight, right? Because alcohol dehydrogenase breaks it to acetaldehyde, right? The first of these aldehydes. And then there is a very important enzyme called aldehyde dehydrogenase 2 that clears it. So it doesn't hang around in you for a long time, okay? Because it's very toxic. So what we did was we knocked out the enzyme that bioinactivates acetaldehyde in a mouse. And this mouse doesn't have a phenotype. So why do you think this mouse doesn't have a phenotype? What observation that is very common to you tells you that these mice would not have a phenotype? I'm turning to the South Korean gentleman at the end. Right, how many relatives do you have? How many of them don't touch alcohol at all?
None of them.
They all drink alcohol. Do you have friends who go bright red with alcohol?
Yeah.
So actually, your friends have a mutation of this gene, right? So each time they drink alcohol, that redness is because of the accumulation of acetaldehyde, right? So it's actually a warning sign that that acetaldehyde is causing damage to you. But our hypothesis is that part of that damage is DNA damage. And our repair pathway is what's fixing that, okay? Right? So we do a very simple genetic test. We take a mouse that lacks this, and this mouse is fine because there's a backup to it. And we now cross it with a mouse that lacks a repair pathway. Now, what I need to tell you is actually mouse knockouts, unusually in this genetic condition, don't look like the human disease at all. And it's been a big mystery in the field, okay? And that will be apparent to you later on as my talk progresses. It's because the first part of the equation, which is to clear the aldehydes, is so efficient in that. So what happens when you combine the two genetic mutations, okay? Do you follow that? Simple genetic test, okay? And this is how you do it. You cross through Mendelian segregation this configuration, and this should give you 25% double mutant mice, and you get none. That is the most fundamental genetic interaction because that's synthetic lethality, that's death. What this immediately tells us is that if you take away the pathway that inactivates the natural aldehyde in you, it causes DNA damage. And if you take the one DNA repair pathway that fixes that damage, then you get lethal DNA damage and an organism can't form. Okay? There is an unusual twist to this genetics because, in fact, what we discovered was something highly unusual. And you won't see this, but I'm sure it'll be a very common feature of human genetics in the future. You never think, you think when you talk about the segregation of genes, you entirely look at the segregation of genes out of a cross, right? You never think what maternal genotype's impact on the fetus is, right? And we discovered that if a mother lacked the capacity to break down acetaldehyde, she never gave rise to double mutant mice, right? Whereas if the mother carried one good copy, she cleared it and the fetus was born.
She cleared it through the placenta.
We don't know how it's occurring, and it's a big mystery, and we've just written a paper in Molecular Cell which you can read if you want to, if you're interested in, that attempts to explain it. But this is known as a maternal lethal zygotic effect. Very few gene classes that exist have been documented in this, but it's probably much more prevalent. But this has enormous implications in your South Korean colleagues' population should a woman drink alcohol in the first trimester, yeah? And I'll come back to that in a minute. Okay, so this is our interpretation of what we think is happening. You all with me? Follow me? So what we know is in our attempt to make a mouse that lacks catabolism and DNA repair, if we get that mouse to be born in a mother that carries one allele of the enzyme that clears the toxin, the embryo is born. If, however, we give it, we try and make it in a mouse that can't clear the toxin, the embryo is killed. What it tells us is a very fundamental and important fact that somewhere during normal pregnancy, metabolism unleashes this aldehyde, and it's got to be cleared by the mother. And we're trying to figure out where it's coming from and how exactly it's dealt with. Okay? Okay, so obviously now you want to know what happens to the fate of the double mutant mice when they're born, yeah? Okay? And remember I told you that the Fanconi straight knockout mice don't show a phenotype, yeah? So what you see here is a Kaplan-Meier curve. We take a cohort of mice and you follow them in a cage. And what you will see in the purple line is that these are the double mutant mice, okay? And they rapidly die. And they die because when you look at their blood, does anyone know what blood looks like? So that's what blood looks like. These are red cells and that's white blood cells, right? You can see immediately if you look at the blood of these mice, it's shot through with these white blood cells. So that's acute leukemia, okay? So that's what happens to these mice. So they get the key feature of the human disease. So if you don't fix aldehyde-mediated DNA damage in a mouse, you reveal malignancy, okay? So actually this work explained two important phenomena, which is why in this disease, what drives the developmental defects, and what leads to the predisposition to leukemia. But people in this field, which has been there for about 35 years, was very skeptical of our work because we didn't explain the main key feature, which is why the bone marrow would fail in the first place. And many people had speculated that there's something that happens in the bone marrow which is peculiar in this disease. So we needed to nail that. And actually we have two problems. One is, what I've given you is an entirely genetic argument. I've taken a gene away and in a combination and largely accepted as a fact that it deals with this factor. So we had to nail the substrate issue and then also prove its specificity and relate it to blood. Okay? So the concept of the experiment that I'm going to show you came, I taste a lot of wine, so that's actually much more famous in Cambridge for tasting wine than anything else. So actually this is a mouse in a wine cellar in Burgundy where this family has been keeping alcoholic mice for many generations. This mouse comes along and it eats food and then it runs along here and then drinks a sip from a €50 glass of wine up here. Okay? So the experiment here is we take our double mutant mice and we expose them to ethanol, yeah? And the logic here is very straightforward, yeah? If they don't have this enzyme, the acetaldehyde should accumulate, right? And this is where it relates to you because actually that deficiency of this enzyme is the basis of the Asian alcohol flushing mutation, okay? 250 million people carry this genetic defect. So if the story I'm telling you, the first tier of control to protect you from reactive metabolites is a very common genetic trait in man, right? The second leg you got to stand on to protect you from these is extremely rare in man, so 1 in 100,000 births. Okay? Okay, so here's the experiment. We do 2 protocols. The first I call the happy hour protocol. So in a happy hour you go there and you binge drink, don't you? Okay? So we take a mouse which would carry a cross, which would carry a double mutant in a heterozygous ALDH2 mother. The fetuses should be viable. And in the first trimester, we give her a single binge drinking dose of ethanol, 1 dose, right? And then we ask what happens to the embryos, yeah? We dissect them out and look at them. And so what you can see is that if we look at the frequency, if we give a saline control, it's fine, yeah? The embryos are born, slightly lower Mendelian ratio, but they can be seen and they're intact. But if we give ethanol, they're wiped out, right? And 6 out of 132 embryos make it through this window, carrying the double mutant phenotype. But if you look at them, they're completely shot to pieces. So here's what a wild-type embryo would look like in this setting, in that same littermate. But one that carries a dual control mechanism either in this instance, completely lacks an eye or is complete exposure to the head. Okay? Now, we're very excited about this result because, as you know, when you pick up a bottle of alcohol, at the back it's got a pregnancy warning signal. And that is because we know that alcohol is a teratogen. It damages fetuses, okay? But if you actually go later on, if you're interested and you read about it, the mechanism of how alcohol causes damage to the fetus is completely unknown. The second thing is it's unclear how much alcohol you have to drink and how prevalent alcohol-induced damage is. It's known as the fetal alcohol syndrome. But actually, there is good evidence to suggest that it's much more common than we think it is. And our work shows two things. One is a single dose, large dose of ethanol in the first trimester of pregnancy is really potently damaging. And the second thing is our work shows that at least an aspect of how the fetal alcohol syndrome arises is due to irreversible genetic damage to the fetus through DNA damage. We did a second protocol. I call that the Ibiza protocol because many of you will go to the south of Spain and instead of drinking water, you go onto a continuous alcohol supply. Yeah? Yeah, and so we took, now in this instance, we take mice that are double mutant and controls, okay? And instead of water, we give them water plus 15% and 20% ethanol for 10 days, okay? Sounds like a party, doesn't it? And then at the end of this, we sacrifice them. And the mice actually looked fine, but the real clue came when we looked at the bone marrow. So you know blood is made in the bone marrow. So Here is a wild-type mouse. Here's the bone, and this is where blood is made. This is the blood production unit. And now if you look in the double mutant mice, it's completely obliterated, right? So we've now made the connection between the accumulation of the toxic aldehyde and the attrition of the bone marrow, right? And it's a very profound experiment because actually, when we were analyzing this experiment, it struck us that this is a bit like taking a mouse and irradiating it. So each time you have a big drink, you know, you may be really subjecting yourself to a significant body of DNA damage. Okay? You don't look too terrified. It hasn't changed the way I deal with alcohol. Big criticism of our work. Of course, children with this disease don't drink alcohol. So is there any evidence that these individuals get, you know, these mice get spontaneous bone marrow stem cell deficiency? Yeah? Okay? So we went back in a second paper to really figure out where, so blood is made in a hierarchical way with the stem cell at the top, and then there are all these precursors of blood at the bottom, yeah? So the stem cell sits at the top of the hierarchy. And for almost 20 years, people knew that the stem cell was very enriched for aldehyde dehydrogenase activity, okay? And it was a big mystery of why this is the case, and many people had speculated about this, right? And in fact, it's used as a marker for stem cells. So what we're able to show, in fact, is that the reason stem cells have this high activity is because of this one enzyme, which quarter of a billion people would be deficient in. And the second thing we went on to show is that if you actually count the number of stem cells in a double mutant mice, yep, without being given ethanol, and I won't go into the nature of the experiment, but it's a quantitative way you assay for the number of stem cells in a mouse. The number is about 1 in 30,000 bone marrow cells or stem cells in a wild-type mouse, but you can see in the double knockout it's 1 in 17 million, right? So that's a very, very big difference. It means that each mouse of our double mutant mice sitting in a cage is entirely producing blood from 2 or 3 stem cells, rather than about 100,000. Okay? And in fact, in our cohort of mice that died of the leukemia, few survived the leukemia, but they all developed spontaneous bone marrow failure. So that's shown over here, yeah? You can see that the blood film here is anemic, and the marrow, sort of having many cells, is a bit like when they were exposed to ethanol. Okay, I'm going to go through this phase because it's a bit aficionado stuff. But really, obviously, the next question you want to know is how can we be certain that any of this is to do with DNA damage, okay? And there's a really cool trick you can do in mice. And when you make red cells, so do red cells have a nucleus? They don't. When they were being made, did they come from a cell that had a nucleus? Must do. Otherwise, they wouldn't have the DNA instruction to make a red cell, yeah? So somewhere in the phase of a red cell, you get rid of the nucleus, you extrude it, okay? But, however, it had been shown that if you take the cell that's making a red cell and you expose it to DNA damage, what actually happens is a broken shard of the chromosome goes off into the cytoplasm and hangs around and forms a micronucleus, okay? And when you kick out the main nucleus, this micronucleus remains behind in the cell, in the red cell, okay? So all you do is you take blood from these mice and you take the red cells and you stain for a marker for DNA, yeah, for the presence of these micronuclei, yeah. And it's a marker of DNA damage. And what you can see is each of these dots quantitates a number of these micronucleated red cells in our mutants. And you can see in the single mutants it's okay, but you can see in the double mutants it's greatly elevated. So it's as though as they're dividing in the bone marrow and producing blood, they're producing precursors of blood with damaged DNA. We combined this experiment with ethanol, and here's the frightening result for your colleagues in Southeast Asia. Because here's with and without and with ethanol, and here's this group, which is just lacking the first protection, quarter per billion people, and you can see that there's a great induction in the amount of damage of DNA in the blood compartment with a single dose of ethanol, okay, in these mice. And obviously in the double mutant it goes way off the scale. So this is an interesting experiment here, because what we've done is we've compared the induction of the DNA damage marker to standard markers of DNA damage. So if you took a mouse and put it into an irradiator, gave it Chernobyl grade irradiation, what's the level of damage? And you can see it's not that far off, yeah? And this is cancer chemotherapy, okay? So the bottom line is, if you're Southeast Asian and you drink, you carry the Flushing mutation, or you binge drink, it is very probable that you're causing significant amount of genetic damage to your blood compartment, okay? The final part of this bit of the story, is, okay, it's all indirect, black dots in red blood cells, that's not DNA, yeah? So can we actually visualize DNA? And this is a very heroic experiment done by my 2 graduate students. And so what they did was they took a mouse and they give the mouse a drug that, that stops the cells in mitosis. And then you take those cells and you get chromosome spreads and then you paint them, right? And then you can look at the shape, the actual integrity of chromosomes directly. And if we do this in our double mutant mice, what we can see is that almost 50% of the metaphases contain chromosome translocations, right? And you can see this visually in that there are all varieties of chromosome damage that you get. So that's what leads to double strand breaks. There are actually many questions that we don't know the complete answers to, and one of them is that. What is it, the nature of, how does it degenerate into a double strand break, and how does that? The final part of our hypothesis, so we'd managed to convince the field that we were probably correct, but then they said, well, you need to show it in humans. And actually we can do that, because as I've told you, that there's a significant proportion of the world that lacks the first tier control, and this rare disease does occur on a worldwide basis. What happens if you go to Japan, where they have very good records in children's hospitals, where a child carries this disease and it's also segregated the ALDH2 mutation, right? Okay, so in fact this was done, and so what's shown here is in this cohort of Japanese Fanconi children, the onset of bone marrow failure with the genotypes, the 3 genotypes, okay? So this is wild type, heterozygous for ALDH2, and that's homozygous, right? So there is a very interesting relationship between man and alcohol, and policyholders and alcohol. But actually one of the biggest problems with the whole alcohol debate is nobody was able to really show why it was a carcinogen, right? There were lots of, there was a lot of indirect evidence to suggest how it might work. And people had speculated about the aldehyde thing, but nobody had been able to prove it. So we hope our work lends some light to it. And in fact, when our article was published in Nature, there was a very nice article written in The Independent on our work, trying to put it in a broader context. Okay, so I'll just quickly finish this part of the talk and then wrap up and tell you one more nice story. This is what we think happens. In you and I, we have this fundamental control mechanism that gets rid of this toxic metabolite. How it comes about naturally in us, where it comes about. And I haven't told you that this is a mitochondrial enzyme, and how it exerts such a profound influence on the nucleus is completely mysterious questions. But should you evade that and it damages DNA, then this repair pathway fixes it. But when you take the 2 control mechanisms away for a blood stem cell, it either dies or it gets to genetic changes that leads to cancer. So it became very important to us, because we're generalists, as biologists, as geneticists, that what we had discovered could be generalized to other aldehydes, okay? Right, so the story I've told you is all about genetic mechanisms to clear acetaldehyde, but there is a much simpler aldehyde that actually is produced by you and me, and that's formaldehyde. And here I've shown it to you in the form you're most familiar with, which is the pickling agent, yeah? And pickling the brain, yeah? Right? But did you know you produce this pickling agent in you? So if I took a blood sample from you, you have a blood formaldehyde level. Did you know that? Okay, and I'll tell you why you have a blood formaldehyde level. Because, do you know histones? Yeah? You've heard of histones, and you've heard of DNA. And you know histones and DNA carry methyl groups on them, yeah? The enzymes that put the methyl groups, and we know that this is very important for the field of epigenetics. But there's also enzymes that reverse that reaction, which are called demethylases, right? And demethylation of DNA releases free formaldehyde, okay? But people in the epigenetics field have never really asked the question, why isn't that formaldehyde toxic to you, right? And so we thought about this and we wanted to generalize our message, but it turns out that there is a completely different enzyme that clears the body of formaldehyde. Okay, it's a completely different mechanism, it's a different class. The only thing similar to ALDH2 is it carries A at the front of it, but its enzymology is completely different, and I've just contrasted it here, yeah? So the question is, what happens when we take out this enzyme in combination with our repair pathway? Is there enough natural formaldehyde produced? So the first time we did this in a cell line, okay? And we found that if we took these two things away, in a simple cell line, it was synthetic lethal. It killed the cell line, okay? So there's enough natural formaldehyde produced from all these transactions biochemically that if you took these two controlled genetic mechanisms away, the cell would die and have lethal, irreversible DNA damage. But to complete this work, we tested it in mice. And for a couple of weeks, we were very worried because the mice were born. So we had a paradox. If you take a cell line, which is a much more simpler system than a mouse, you know, why aren't we seeing lethality? We don't know the answer to that. But is there any evidence that this mechanism operates in a live mouse? Well, you don't need to be a rocket scientist to know that that's a wild-type mouse and this is a double mutant next to it, right next to it, okay? Absolute age match. So it's severely growth retarded. And in fact, it doesn't have a very long life because you see it drops down like a stone, yeah, in green. It rapidly dies. And this in contrast with the system that deals with acetaldehyde. So two important messages. Our story about aldehydes is probably generalizable. And in fact, these are just two aldehydes. There are probably another 18 classes of aldehydes, all dealt with in a unique way in the body that's cleared, okay? And these mice die of bone marrow failure, and they don't have stem cells. And so we think in a stem cell, there are two systems that at least operate, one that clears formaldehyde, one that clears acetaldehyde, and this common DNA repair pathway. I'm going to go a bit fast because I think you'll reach saturation now. You know, obviously the acute anemia does not explain why the mice are severely growth retarded. And in fact, we find they get explosive liver disease because their liver contains these very large nuclei. You see these? So the nuclei have a double mutant compared to the size of the nuclei over here. And that's because the nuclei undergo many rounds of DNA replication because their DNA is glued and they may not be able to resolve it. So it's like copying glued strands, a bunch of glued spaghetti, yeah? And we don't understand why this is occurring and what the basis of it, but a portion of my lab works on this, okay? So I'm just going to wrap up. They get also kidney failure, but this will be too complicated to explain it to you because you'll reach saturation because it's obviously very complicated things happening in this mouse that lacks the formaldehyde system. Can we rescue it? And we did a bone marrow transplant, and in fact we can get it past that lethality window. But then actually all the mice die of liver failure, and they sometimes get cancers. Okay, it's shown over here. The one thing about this enzyme that breaks down formaldehyde is that in the liver it's present in the cytoplasm and, as you can see, in the nucleus. Okay, so maybe the enzyme tracks the source of formaldehyde. If it's coming from histone demethylation, it might be put in the nucleus. If it's coming from the outside, it may be put in the cytoplasm. And this is most strikingly seen in the kidney. So if you take a kidney, this is a nephron, right? You know, and you mark it out in these two sections, okay? And what you can see is the antibodies the formaldehyde enzyme is shown here in green. What you can see in many of the red staining cells, yeah, in this section of the kidney, the enzyme is entirely in the nucleus. But in other parts of the kidney, it's exactly in the opposite compartment. And we're very excited about this result because this suggests to us that cells might have a formaldehyde sensing mechanism. So it puts the enzyme in the right place from where the enzyme is coming from. That's obviously a speculation, but it would lead us to exciting new biology, and we're trying to prove that. And the very worrying thing right now is a lot of pharma companies are making inhibitors of this enzyme, right? And the reason is it has another substrate, which is nitric oxide, the gas, right? And nitric oxide causes blood vessels to dilate and it causes tracheas to dilate. So it would be a very attractive drug to give to people, who've got asthma. And inhibitors of this enzyme are already in phase 1 clinical trials. And I think that's a very fatal thing to do because it's leaving you unprotected against natural formaldehyde. Okay? And actually, it's a very curious thing. I felt it pricked my conscience so much that I actually wrote to this company and told them about our data, but they never communicated back to us. So, Okay, I'm just going to wrap up and what I think how studying a very, very rare and unusual disease, we believe we've discovered a fundamental physiological system that protects you against something that's produced within you as a cost of metabolism. So natural metabolism produces this molecule. There's a whole battery of these enzymes that clear these. And I've only told you about one in a class of 18 in fact, of the ALDH2 family. So you have another 17 proteins that also break down aldehydes, right? And there are another 8 in the ADH5 class. So obviously evolution has devoted a lot of these enzymes to clear these reactive molecules. And I've only really told you about 2 of them, If you lack this genetic control, as you might do in the Asian flusher, or you overwhelm it by going on a bender, you damage your DNA, and that's when you're reliant on this system. And we think dysregulation of this dual protection mechanism may account for fetal attrition and the fetal alcohol syndrome, or the defects you see in Fanconi's anemia with development. We know it's pretty fundamental for the production of blood. And finally, we know that it's a very efficient way to protect you against cancer. Okay. This work was actually all done by graduate students. I drink a lot of wine, so they obviously don't believe in what they do. But those 3 guys are all PhD students in my lab, and all had Nature papers in the first 3 years of their PhDs. And the work on the alcohol-maternal relationship, which I have not told you about, was done by a very good lady PhD student in my lab. But I haven't told you about that. And some of the formaldehyde work was done by a postdoc who's rather sanctimoniously pointing to a Coke bottle. And that's it.